Welcome, everyone, to our lovely campus, and welcome back to some of you. We're delighted to see all the alums here, so hopefully you saw all the wonderful changes and all the new buildings that seem to have grown on our campus that maybe you didn't see before. I'm Julia Greer, and I'm the Fletcher Jones Foundation Director of the Kavli Nanoscience Institute here at Caltech. Hopefully you've had a chance to see it. I'm also a professor of all things that begin with an M, but that's not important right now. My job here is to introduce today's speaker, Assistant Professor of Biology and Bioengineering, Dr. Rebecca Voorhees, who is the 6th winner of the KNI-Wheatley Scholarship in Nanoscience. This is a very special program that was created through the generous support of Caltech's distinguished alumnus, Chuck Wheatley, and his late wife, Judy Wheatley. The KNI-Wheatley Scholarship supports outstanding tenure-track Caltech faculty members in their pursuit of novel nanoscale research. A little bit of information and background on our outstanding speaker today. Rebecca performed her graduate work in Venki Ramakrishnan's lab in the Medical Research Council Lab of Molecular Biology at Cambridge, not the one on the East Coast, but Cambridge University in England, where she studied molecular mechanism of protein synthesis using X-ray crystallography. For her postdoctoral studies, Rebecca stayed at the same university and joined the lab of Professor Mano Hedge, where she focused on using single-particle cryo-electron microscopy to study how secreted and integral membrane proteins are specifically targeted to the endoplasmic reticulum. Hopefully I'm pronouncing things correctly. Rebecca moved to Caltech in July 2017, where she joined the division of BBE, as we know it, of Biology and Bioengineering, and her career just was launched immediately as she was recognized by the Searle Foundation. She became the Pew Stewart Scholar and the Heritage Medical Research Institute Fellow. In 2019, she was the recipient of a very prestigious award. It's the NIH Director's New Innovator Award, and in 2023, she was named one of the 31 inaugural Freeman Hrabowski Scholar at the Harvard Hughes Medical Institute. Today, well, you've been staring at this title for a while, so today Rebecca will talk about protein complexes as the nanomachines of the human body, their manufacture, assembly, and destruction in health and disease. Her lab studies the complex process of protein development to better understand how healthy cells function in an effort to create more effective therapeutics for human life. After her talk, Rebecca will stay to answer some questions, so please wait for the microphone to come to you so that our audience that's on Zoom or that's going to watch the lecture after will be able to hear the questions. So with that, please join me in welcoming the 2024 KNI-Wheatley Scholar, Professor Rebecca Voorhees.
All right, so I'll start just by thanking Julia, of course, and saying a special thank you. Can you hear me? The mic? No. There's supposed to be a body mic. Is it working? No. If not, I'll stand behind the lectern. So I wanted to start, of course, by thanking the Kavli Institute and the Wheatley family, of course, for the support, not only for the opportunity to be here and tell you a little bit about what our lab does, but also because it's just this really great privilege to have unrestricted funding to do the research in our lab that we are most excited about and we think will be most transformative for our field. And that's what this scholarship has allowed us, at least in part, to do. And so it's something that I think is a really special and unique thing that I value about both the scholarship and then being at Caltech in general, is that we have these kinds of opportunities to do research that we're just excited about, that maybe some federal agency wouldn't be so excited about. So if you're alumni, that's my plug for these kinds of scholarships and fellowships. And so the other thing I want to say is thank you for choosing me. So I have kind of a secret that maybe we can keep between us and then all the people who are going to watch this on YouTube afterwards, is that, I'm kind of a weird person to pick for a nanoscience scholarship or program. I'm not an engineer, I'm not a material scientist. At the end of the day, I'm really just a biologist And so our lab is interested in understanding the nanomachines, is what I've called them, but they're really the molecular machines that underlie cellular life, that allow our cells in our body, all 47 trillion of them, to function and carry out the day-to-day activities that support human life. And so we're really interested in understanding both how these work in healthy and normal cells, and then also how they break in the case of disease. And so hopefully by understanding them at the nanoscale, then we can actually understand, have the information we need to correct them when they fail. And so when I was thinking about how to introduce this topic, I was assuming that most of the alumni probably didn't study biology. Is that true? How many people studied biology? All right, there's a few, that's good. But I figured I'd try to kind of ground us in something that we could all appreciate. And, caltech is just famous, really infamous for its rigorous curriculum, absolutely impossible academic standards, and terrible sports teams, right? And so I figured that for that type of illustrious, very educated audience, I would pick something that we could all appreciate, which was the children's picture book. And I like this because it's this really kind of easy visual representation of the infrastructure that we know that supports our towns and our cities and our neighborhoods. And hopefully this is something that some of you are familiar with, those of you who have kids or grandkids. Richard Scarry, Busy, Busy Town, one of my kids' favorite books. And the reason that I use this is because it's a way for me to explain what our lab is interested in doing. And we want to understand how all of the infrastructure that supports human cells, that's analogous to really what exists in cities, how it works, what it looks like, and what happens when it fails. And so just to make this a little bit more concrete, these are things themes that I'm going to come back to throughout the talk. So the first is this idea of compartmentalization. And in a cell, that means these different regions that can carry out different functions. In a city, these are different buildings, maybe, different places where things happen. And just like in a town, the things that have to be at a school are different than the things that need to be at a library or that need to be in the town hall or the courthouse or whatever it may be. And if there's mixing of these things, they end up in the wrong place, the kindergartners are running the courthouse, you've got a problem. And it's the same thing in a cell. So making sure the right things are in the right place really matters to allow cells to work. The second thing is that all human cells have this really highly developed transportation architecture or infrastructure that actually moves things to the right place and between these different compartments. And it's just like, you get in a taxi, or probably not a taxi anymore, an Uber, and you give it an address, and hopefully they take you to the right place. Now, I found out on my last trip that's not always true. Sometimes they drop you off at the wrong place. And just like in a city or in a cell, if that happens over and over and over again, things just don't work. And then finally, and perhaps most importantly, is that there have to be ways to clean up mistakes or problems. And so in a city, of course, this is our sort of garbage trucks or recycling centers. And just as if you imagine, garbage is piling up in the streets or no one's emptying the garbage can in school, it might make it difficult for you to get to work or your kids to learn things in their classrooms. This is true in a cell too. So if you accumulate too much garbage, then the cell can't function. And so there's ways to actually get rid of it. And so really at the end of the day, what our goal is to try to understand how these cellular molecular machines work that really control the movement of things between these different compartments in order to maintain this architecture and organization of the cell. And so I kind of move away from the pictures here and actually move to the things, actual images of cells. So of course now we're not talking about buildings, we're talking about organelles. And then this is what we're actually looking at inside a cell. So this is an electron micrograph from a eukaryotic cell. It's actually from a guinea pig, but it looks just like a human cell might look. And you can see that it's really crowded. So there's lots of things going on, but there's still these really exquisite compartmentalization and organization. So each one of these organelles is surrounded by what's called a membrane, which basically keeps everything in and separates it from the outside. And so it's really within these individual compartments, these organelles, that you have to make sure the right things get sorted and they end up in the right place. And so, in the city analogy, we were talking about maybe people or books or, I don't know, pencils, school supplies ending up in the right place. In the cell, we're almost always thinking about, at least in our lab, about proteins. And so proteins are the sort of workhorses of the cell. They carry out all of the processes that I've talked about already, moving things around, getting rid of things, and they also transport themselves. And so I'm going to tell you a little bit about how some of these proteins work, and in particular, how they actually sort proteins between compartments. And so we're really interested in not only how these proteins are made, which mostly start on this nanomachine called the ribosome. I'm not going to get to talk about today, but I studied a lot as a trainee. And then they have to be sorted to the right place, and this involves these sort of cellular transport factors like buses or taxis. They kind of operate in the same way. They use a molecular address and sort of a molecular mail truck to move things to the right compartment. And then, of course, there's also a way to get rid of cellular junk. And I will say that this is kind of where my analogy breaks down, because, in a city, if you end up in the wrong place, you just try again and hopefully end up in the right place a second time. The reality is in a cell, and I'll come back to this, is most often when something's in the wrong place or a protein fails to mature properly, we actually just chop it up and degrade it. And so we don't do that in cities, but I'll point that out later. This is important for later in the talk. So the analogy is not perfect, okay? But that is really the idea. So everything we're trying to do in the lab is to understand what I like to call it the molecular logic of how proteins are made and moved around the cell. And the idea is to understand this at a molecular level of detail, that not only we can change where proteins go, so maybe they go to a different place or they're at a different level, so they accumulate maybe, but also so that we can manipulate how cells respond to their environment. And the reason that's important is because if we can change where proteins go and we change how cells react, then we have a chance to actually influence something like organismal health and disease. And so we know now that the organization of the cell and making sure that it's really exquisitely compartmentalized and that proteins get to the right place at the right time has to happen with a very high degree of accuracy. And what's kind of remarkable is this happens on a massive scale. So the average human cell is going to make something like 10 to 20 million proteins a minute. And so that means you have 47 trillion cells in your body, and every minute you have 20 million new proteins that have to be sorted correctly. And this has to happen with this just crazy high degree of accuracy. We know this because when it doesn't, you get disease. And to make this more concrete, I want to show these videos. Now, they're a little bit old. They were taken when I was a trainee, so you do better on your iPhone for sure now. But on the left here is a healthy mouse. And on the right is a mouse where one of the 20,000 proteins that are encoded in the human genome has just one mutation. And so this one protein, this mutation, all it does is it means that this protein ends up in the wrong place twofold more often than normal. So it goes to the library instead of the school, or whatever the analogy might be. And what you can see is even with just this really subtle breakdown of cellular organization, at the organismal level, this mouse begins to display what we call the classical phenotypes of neurodegenerative disease. It's hard to say that the mouse actually has neurodegeneration because mice don't really have neurodegeneration per se, but they have some of the similar characteristics you see in human patients. And so that's part of why we think it really matters to understand these processes at the molecular level. And so one of the things I'm going to tell you about is some of the research we've been doing over the last few years at Caltech that has to directly do with how proteins are sorted between compartments. I'm going to switch to using this sort of coloring book diagrams of cellular organelles. They're not as pretty as the real thing, but they're a little easier to visualize. And what I'm going to talk about today are really what I like to think of as sort of the molecular gates that sit at the entrances to different organelles. And what I mean by that is I was flying in from DC last night, actually, and I parked the car at LAX, and you get to the exit of the parking garage, and you scan your QR code, and you hope, you cross your fingers that it opens. And it did, because that's why I'm here. But this is basically what these things are. They're molecular gates. They sit at the entrance of different compartments, and not only do they let things in and out, but they do it in a selective way. So it's not just something that's open all the time, it's that you actually have to scan a QR code or a parking pass or whatever it is to get in. And only those things that have the right QR code get access to a compartment. And that's how you get protein sorting. And so I'm going to tell you about what we've learned about some of these different molecular gates at different organelles. So I'm going to start in my favorite organelle. This is the endoplasmic reticulum. And I've worked in this area for a long time. And I first got interested in it because a very large fraction of proteins actually have to passage through the endoplasmic reticulum. About a third of proteins in a human cell go through this organelle. And it's not just a lot of proteins, but they're really important proteins. So anything involved in cell-to-cell signaling, which basically means how cells either communicate with themselves or how cells communicate with their environment, has to passage through the endoplasmic reticulum. So these are things that have to get to the outside of the cell and either be secreted so that they can interact with other things in the body, or things that sit in the very outside of our cell called the plasma membrane that really sense and communicate information to the rest of the cell. So hopefully these are things you've heard of, antibodies that recognize invaders in our blood, digestive enzymes, transporters, some types of hormones. And then also I like to point out in the post-pandemic era that this includes viruses as well. It's not just these sort of host proteins that have to get into the endoplasmic reticulum, but also viral proteins that hijack all the machinery I'm going to talk about to make their own viruses and then ultimately exit our cells so they can then infect more cells and propagate and so on. In addition to these being important proteins, they're major drug targets. So about 50% of FDA-approved drugs target a protein that has to go through the endoplasmic reticulum. So this includes things like, if you take an antihistamine, if you take something to modulate your pain receptors, your blood pressure, any of those, and many, many others. They all regulate these proteins. And so we really care about understanding both how these are made, how you control their levels, and then where they are. Because if you want to drug them, you better have some information about how all of that's working. And so we started with this question. So how do these proteins get into the endoplasmic reticulum? And how do they do this selectively? How do you pick the right ones that should get there and reject the ones that shouldn't? And one of the first things we started working on was one of these molecular gates. It's called, the name isn't so important, but I'll give you it just for sort of simplicity. It's called the ER membrane protein complex, or the EMC for short. And it sits at the entrance to the endoplasmic reticulum. So ER is a short version of endoplasmic reticulum, not an emergency room ER, but sometimes you'll see it on my slide just because the titles didn't fit. So we knew when we started working on this that the EMC was this really important molecular machine because a number of different physiologically important proteins relied on it to get access to the endoplasmic reticulum. This includes things like the opioid receptor, histamine receptors, and many viral proteins as well. And so we knew it was important, but we had no idea how it worked. And so one of the ways to try to figure out how it worked, we thought, was to actually look at it at a molecular level, to understand how it functions, and then to maybe learn something about how it picks its clients. And so one of the strategies that we use in our lab to study proteins like this, especially these sort of larger nanomachines, is a strategy called cryo-electron microscopy, or single particle cryo-electron microscopy, and I'll explain why you use that single particle name in a couple of slides. And we have a fantastic facility. I don't know if you get to go on any tours, but an amazing facility in the sub-basement of the BI now that's been set up. It's not that old, maybe 8-9 years ago. And so we can do all of this at Caltech now, which is pretty amazing. And so the basic idea is you purify a protein or a protein complex, and then you put it onto a support. And this is an electron microscopy grid. These are about 1/2 centimeter in diameter, and they look something like this. And so what you're seeing here, these crossbars are metal crossbars. They're just supports, so they can be made out of copper or gold in some cases. And then inside is this sort of holy carbon film. So it's a carbon film that has holes punched out of it. And inside these holes, if you've done it correctly, you'll get your particles suspended. And after they're suspended in the holes, then we flash freeze them so that they're suspended in this very thin layer of vitreous ice. And so it's going to turn to the side, and you can kind of see what this looks like. And so for an ideal perfect sample, which of course never exists in the lab, they never look like this, but sometimes, maybe one day, you get samples that are perfectly suspended in the center of this sort of very thin layer of vitreous ice. This allows them, when they're cryoprotected and cooled, this allows us to actually put them in an electron microscope and shoot them with an electron beam. And then we collect hundreds, thousands, many, in some cases millions of images now. And these images are each of an individual particle. And so that's why it's called single particle cryo-EM. So we can actually look at every single one of these particles on its own. And this is really exciting from someone, so I was trained as an X-ray crystallographer, and so to do X-ray crystallography, you need a lattice of particles or chemicals or whatever it is that are all identical and kind of arranged in a very particular order. The magic of single particle cryo-EM is we don't need that anymore. And so we can look at samples that have much more heterogeneity, maybe movement, and try to understand more about our particles using these kinds of approaches. The images you get are a two-dimensional projection of a three-dimensional object, so kind of like an X-ray of your hand, for example. And so then you computationally reconstruct the three-dimensional density. So you take each one of these images, you hope that they are sort of uniformly represented around the sphere. And if they are, then you can get a high-resolution image of this sort of three-dimensional molecule that must have created these two-dimensional projections. And when this works well, you get a structure. And so when we started working on this molecular gate, the EMC, a really fantastic group in my lab. So Gio is a chemistry PhD student, Kurt was a staff scientist, and Tino is a postdoc. He's now running his own group at Stanford. And so I will say one of the amazing and really kind of special things about being at Caltech is the spectacular trainees that we have, PhD students, postdocs, and staff. It's just amazing. And so what these three did is they applied this entire pipeline to this complex, the EMC, from human cells. And they got a map that looks something like this. So the gray basically tells you where all the atoms are in the structure. And then we can color it based on what we know about the complex. So we know that the EMC has 9 subunits. I'm not going to talk to you about this, but they have to be put together kind of like a Lego model. The way these complexes are built is something that I think is really interesting to study too, and we work on. But what we were able to learn from this structure is a little bit about how this complex actually works. And so one of the first things they did, they brought in another graduate student, Masami, who's a biology PhD student, and they decided to try to figure out which part of this complex actually functions as this sort of gating mechanism into the endoplasmic reticulum. And so I'm going to show you, it's this little region here that kind of sits in the middle of the complex. And if I show you now, this is a slightly different representation. So it's based on what's called electrostatic potential, which is just essentially the charge around the surface of the molecule. Positive charge is in blue, negative charge up here is in red, and then things that are uncharged are in white. And what you can hopefully appreciate is that we now know that this has a sort of positively charged filter into this compartment. And through some really painstaking experiments, I'm summarizing with a super lame cartoon. I hope Masami's not watching. I think she's on vacation, so I'm okay. We know that substrates actually have to directly go through this positively charged filter. And so if we go back to our cartoons now, we know that we have a gate into the ER that has this positively charged filter. And we can show that substrates that are supposed to get into the endoplasmic reticulum, they actually go through this gate. And that makes sense, because most substrates that go to the endoplasmic reticulum, they're enriched for negative charges or neutral charges in the regions that actually have to interact with this filter. But what was unexpected, at least to us, is that this also has a role in selectivity, because proteins that have to go to other places, so like the mitochondria or another organelle called the peroxisome, they tend to have positive charges in the regions that would have to pass through this filter. And so you can imagine that you would get some sort of repulsion, like putting together two sides of a magnet. And that means that these proteins actually can't get into the endoplasmic reticulum. There's some sort of specificity. They don't have the right QR code to open the gate. And we know that this is the case because we can actually play with the properties of the filter and change what happens. And this is kind of back to this idea I told you that we really want to be able to manipulate protein flux, not just understand it, but change it. And so if we make this filter negatively charged now, we relieve this repulsion, and all of a sudden you can get proteins into the wrong place. And so this means that what actually happens is that more of these proteins end up the endoplasmic reticulum, and you actually see less in the correct place, like the mitochondria. And so this is a really nice example, I think, of understanding the molecular basis or the chemistry of how these types of nanomachines actually work allows you to manipulate where proteins go in the cell in a sort of rational way. And it also tells us something interesting about these molecular gates. We understand something about the molecular QR code that allows some proteins access to this compartment, but rejects others. And so that tells us something about how the cell is maintaining this compartmental organization and architecture in order to preserve what we like to call cellular homeostasis. So basically the balance of all proteins in the right place and allow the cell to actually function. So we'd worked a lot on the endoplasmic reticulum, but a couple of years ago, we realized that, while we'd been thinking a lot about how mitochondrial proteins get to the wrong place in the ER, we didn't know very much about how they get to the right place. And that was kind of surprising to me because we know and have known for probably 40 or 50 years now that these proteins that have to get to the mitochondria, they do really important things for cells and for organisms. And so mitochondrial proteins, they do things for human cells, like they recognize cellular invaders. They control this pathway called programmed cell death or apoptosis, which I'll come back to. And because of that, when these proteins don't get to the right place, or they're not made correctly, or they end up in the wrong place, you get these diseases. So things that are sort of major societal problems, Alzheimer's disease, Parkinson's disease, obesity, cardiovascular disease, these are all, at least in part, associated with dysregulation of proteins at the mitochondria. And yet, we had no idea how these proteins get there in the first place. And that was a little bit strange, but I think part of it had to do with the evolution of mitochondria. And I thought this would be kind of interesting to go through maybe. And that's because mitochondria evolved from an autonomous bacterium. And so basically the idea is that about 1.5 billion years ago, there was a very simple organism that basically had a cell membrane, DNA, so some sort of nucleic acid, genetic information, and it was engulfed by a second simple cell. And most of the time when this happened in evolutionary history, this wasn't very useful, and then this sort of organism would be lost. But in this case, you actually had this sort of mutually beneficial relationship, so a symbiotic relationship, just like we talk about with organisms or species, animals. And so this organism, which is basically similar to a modern-day bacterium, was retained in the cell. It helps the cell avoid invaders. It provides energy. And then there was this exchange between this organism and the cell nucleus, where a lot of the genetic information became housed in the cell nucleus. And this is basically this endosymbiotic theorem of how you get mitochondria. So you go from an autonomous bacteria to an endosymbiotic organism to an organelle. And I think that's why we didn't understand how these proteins got to mitochondria, because they weren't there in the original bacteria. So when mitochondria had to go through this evolutionary process, they had to figure out a way to communicate with the cell, because it's only symbiotic if you can actually talk to each other and communicate the cellular environment, the needs, energetic needs of the cell, and so on. And most of this communication is mediated by proteins that sit in what's called the outer membrane. So mitochondria have two membranes. I'm only going to talk about this one, this outer membrane today. And it has kind of two families of proteins. So one form these sort of barrels, look like this. And those are commonly found in the outer membrane of bacteria. So they probably would have been there in this ancient, our ancestral bacterium that was engulfed by that cell 1.5 billion years ago. And so we actually know a lot about how this pathway works, how these proteins get in, and what are the molecular gates that allow them in the first place. But conversely, all of the proteins I talked to you about at the beginning that do these really important things for human cells, they actually don't have this barrel shape. They have what's called these alpha helical transmembrane domains that I represent as these sort of cylinders, these skinny tubes. And so they look a bit different, and they aren't commonly found in the outer membrane of bacteria. And I say commonly because I was talking about this work a couple weeks ago, and I said never, which was always a mistake in science. You never say never. Because then I say never, and I see this hand kind of in the back of the room that goes up. And, I say, yes. And there's one alpha helical protein, the outer membrane of some bacterium somewhere in the world. But typically, they just aren't there. And so what that means is that these had to evolve after this endosymbiotic event, and the machinery, the molecular gates, these molecular machines that put them there, had to evolve after that as well. And I think for that reason, it's been a really hard problem to crack for the field. So we've known that this must have been happening for decades, and we just had no idea how it worked. And so this was a problem our lab got really interested in a few years ago. And again, we had this amazing group of people. Taylor is a chemistry PhD student. Allison was a postdoc. And then Alina was a joint postdoc between my lab at Caltech and Jonathan Weissman's lab at MIT. So this was a collaborative project. And so they just asked, well, can we find a molecular gate or the molecular gate that actually puts these proteins into mitochondria, that allows them access to this cellular compartment? And so the strategy to do this is a little bit different than what I told you, because before we knew what we were looking for. We knew the answer, we just wanted to understand how it worked. But now we're looking for a new pathway. And so we used a strategy, a genetic strategy. It's a genome-wide CRISPR screening strategy that allows us to basically brute force look across the entire human genome, get rid of one protein, one of each of the 20,000 genes, one at a time, and see which ones affect our process of interest, so how proteins get to mitochondria. We did this with a reporter. So we took one of these proteins that get to mitochondria, we put a fluorescent protein on them that would allow us to detect when they got to the right place. And then we got rid of each protein in the human genome one by one. Luckily, we don't have to do this one by one. It's not 20,000 different experiments. We do it pooled in this sort of big flasks with billions and billions of cells. And when you do this, then you can look for things that affect our reporter. We summarize the data like this. So each one of these little gray dots is one of 20,000 genes in the human genome. And then in this case, you have one hit way off on its own. And it was this protein called MITch2. And we could show that if you get rid of MITch2 in many different ways, we did lots of different experiments I'm not going to talk about, that proteins cannot get into the outer membrane. So you get rid of MITch2, you lose your molecular gate into mitochondria. And the reason that we were excited about this, and I think this was interesting, particularly interesting to us at the time, is that MITch2 had actually been studied for decades. People knew it was really important in human disease, but they had no idea why or what it did. Just that when you messed with it, people and organisms get sick. And they get sick in all these interesting ways. So, for example, if you have higher levels of MITch2 expression just naturally, there's sort of variation across the population, you're more likely to get Alzheimer's disease. We know these from these genetic association studies. If you have differences in MITch2 activity, you're also more likely to get cardiovascular disease, obesity, and this is going to be something I'm going to come back to, but MITch2 levels are markedly altered in many different types of cancers, both solid tumors and many blood cancers, lymphomas and leukemias. And what was sort of interesting is that the only thing that all of these things had in common, in addition to some defects that actually relate to mitochondrial function, is that they have to do with dysregulation of mitochondrial proteins, and in particular those proteins that sit in the outer membrane. And so that was kind of interesting. And we've been able to show now why that is. We think that MITch2 is this molecular gate. It sits at the entrance to mitochondria, and it selects which proteins get in and which ones don't. And I'm not going to go through all the data that suggests that that's the case, but I'm just going to spend the sort of rest of my time telling you why you should care about that and why we think that this is important. And so the group in the lab now has been focusing on answering these two major questions. First, understanding the conservation, the evolutionary conservation of molecular gates like MITch2. So how many species have them and why? Why are they conserved in some species and not others? And then understanding its role in disease. So I'll start by telling you a little bit about its conservation. So what's sort of interesting is that we think that there are versions of MITch2. So there's a type of MITch2 in all species in the animal kingdom. And so that means in the outer membrane of mitochondria, in everything from choanoflagellates, where there are these sort of single-celled organisms, all the way up to humans and mammals, there is a version of MITch2 that sits in the outer membrane of mitochondria. And what's crazy to me, I mean, absolutely crazy, the students suggested this experiment. I told them it wouldn't work, and of course they never listened to me, and so they did it, and it worked. But what's amazing is that the evolution is such that you can take one of these MITch2s from a single-celled organism, and you can put it into a human cell. You can give it human clients, and it'll still put them into the mitochondria selectively. So not just everything, it's not an open gate, it is an active gate that seems to be able to recognize this sort of human QR code, even across all of this evolutionary time, which is kind of amazing to me. We don't have a real structure of what MITch2 looks like, but this is a model that we've been able to predict. And again, I'm showing it based on sort of the charged potential of the surface. And so hopefully what you can see is that just like in the ER, this gate also has this sort of charged filter mechanism at the entrance to the mitochondria. And we know that this is important because if we change it, so we make it more negative or we make it more positive, then we can make the molecular gate more or less active. And I kind of think of that as like how fast that arm goes up when you're going in a parking lot. So more active, it goes up much more quickly, and one that is completely inactive, it always stays closed. It doesn't matter if you have the right QR code. And so what that tells us is that I think this is a really beautiful example of how biology uses the same solution to problems over and over again, even evolutionarily unrelated molecules. And so I told you about this complex at the ER membrane. This is at mitochondria. And then these are other gates that sit at either archaeal membranes or bacterial membranes. And so what hopefully you can see is that they all use these charged filters to actually select their substrates. But also hopefully what you can appreciate is they all look a little different. There are different widths, different lengths, different amount of charge, different types of charge. And we think that's part of how you get selectivity between membranes, is that this is precisely tuned to the right set of substrates, so you pick the right ones and hopefully reject the wrong ones. And so I think this is sort of just a fundamental feature of all of these molecular gates that sit at the entrance to different organelles, and tells us something about how you get this really exquisite level of protein sorting between compartments. And then the second thing that I think we've been really excited about, and I'm going to finish on, is that we think that understanding what MITch2 is actually doing gives us this unique opportunity to leverage this information for therapeutic benefit. I already told you that MITch2 is involved in all of these different diseases, and I think we can explain why now. And I think we might begin to have the tools to actually manipulate it in hopefully a therapeutically relevant and useful way. And so one of the things that I've sort of mentioned briefly is that if you have MITch2, everything gets into the mitochondria just fine. If you get rid of it or you make it inactive through mutations, then these substrates don't get to the mitochondria. But they don't just accumulate either. They actually get degraded by the cell, so they're completely removed to protect the homeostasis of the cell. And so this is a really useful property therapeutically, because if you can change MITch2's activity, so you can maybe inactivate it, then not only do you prevent proteins from getting into mitochondria, but you completely get rid of them. And so you can imagine that that could be useful if there's a particular protein that when it's upregulated or when there's too much of it, it leads to disease. But what's sort of special about MITch2 as well is that you can make it more active. And so if you make it more active or you have more of it, you get more of these proteins into mitochondria. So you can actually enhance this effect. And that's somewhat unusual, I would say, for drug targets. And so what we're really interested, and so what this tells us is that MITch2 is really the sort of regulator of the outer membrane of mitochondria. It selects which proteins get there and their levels. And so we're really interested in using this information to ask the question, can we manipulate MITch2 to change how cells respond to their environment? And I'll come back to the list I showed you at the beginning of this section, which is that we know now that many of MITch2's clients, so proteins that rely on it to get into mitochondria and therefore to function, are important in disease. And so one of the first things we targeted was this programmed cell death pathway called apoptosis, which is really important in cancers. And I'll explain why that is. So the word apoptosis comes from the Greek for falling off, like from leaves from a tree. And the idea is basically that if you can cut off the dead branches, so this is the beautiful oak tree that used to be near the library, some of you may remember it from your time at Caltech, and for many, many years, since I was on campus, they were cutting off dead branches in the hopes that they could save the tree. It didn't work in this case, but hopefully apoptosis works most of the time in our bodies. And the idea is you cut off the dead part to protect the whole, so a sacrifice of a portion to protect the organism. And the reason that this is important in cancers is, for example, if you imagine cells in your blood, if one of them is affected either by genetic mutation or the environment, and you get a mutation that's going to cause a cancer or disease, if this cell just dies off, basically nothing happens. You lose one cell. In most cases, except for neurons, you're pretty much fine. You can afford to lose a few cells. However, if apoptosis pathways aren't working, then the cell remains, it becomes mutated. So now it's a cancer cell, and then it can divide and divide again, and then divide again and again and again, and so on. And so you get either a solid tumor or blood cancer, for example, in this example. And so that's why for most cancers, it's absolutely critical to shut down apoptosis, because if that cell had just died, there'd be no problems. And so therapeutically, one of the major goals of cancer treatments is to activate or stimulate apoptosis in tumor cells. And one of the major goals of cancer cells is to prevent apoptosis, to allow them to continue to proliferate. So it's this sort of arms race. And so we're hoping now with this sort of unique insight that MITch2 might regulate apoptosis, that we might be able to actually change how cells respond to these drug treatments. So as sort of first test of this, we took some leukemia cells. We treat them with a drug. It's marketed under the name Gleevec in the United States. It's a frontline leukemia drug. And then we basically look for apoptosis using this sort of dye, fluorescent dye. And so when you actually do this, we can detect how many cells are dyed with our fluorescent molecule. And when you do this with control cells, you get just a tiny bit of apoptosis or cell death. And then if you treat cells with drug, you get a sort of modest stimulation. Leukemia cells that we work with in the lab are highly resistant to these drugs, similar to what happens in patients, but we also see this in the lab, particularly with the strains that we often use. But what was interesting is that if you add more MITch2, so you overexpress it, now you get a little bit more cell death when you treat with drug. It's modest, twofold. But what was interesting to us is that it's not just the amount of MITch2, it's actually how active it is. So in this sort of analogy of the gate, it's how often that gate is going up or how fast it can go up. And so now drug treated is in black and control is in gray. And so if you add inactive MITch2, we basically see no effect. Does that make sense? But what was cool is that if you can make it hyperactive, now you can actually get significant stimulation. And what's interesting here is that you get 5-fold stimulation or so in the presence of drug, but you also get some stimulation in the absence of drug. So Mich2 on its own can actually trigger these cells to undergo apoptosis. All right. And so that's kind of our hope, is that, sorry, is that if we can manipulate Mich2's function, then we can actually do this in a therapeutically meaningful way. And we think that this might be important because, as I mentioned in the beginning, there are many types of cancers where the levels of Mich2 are really altered. And so this is just a bunch of different types of tumors. And in red is the levels of Mich2 in tumors, and in black is a like-to-like comparison with a healthy cell. And so if you look at something like leukemia, which was the cells I was talking about, you have several order of magnitude increase in Mich2. And then if you look at things like lymphomas, you actually see, decrease in Mich2, and this is an increase in Mich2 in tumors. And so you actually see both phenotypes, where you have much more Mich2 or much less, depending on the different types of cancers and sort of the proteins that they express. And so we're very interested in seeing if we can find small molecules that really phenocopy the effect I showed you, where you could add a small molecule and then maybe you could sensitize cells to these drugs and increase apoptosis. And so just to summarize what I've told you in the second part of the talk, we found what we think is the new molecular gate in the mitochondria. It uses this charged filter, like many different kinds of molecular gates, to different compartments. It looks a little bit different than those that are at the ER, and so we think that's probably important for selectivity. And then it also regulates the levels and the types of proteins that get into mitochondria in a way that can affect how cells respond to their environment. And so if we kind of put this all together, really we're sort of slowly inching towards our goal, which is to understand the molecular machines that really control cellular infrastructure, that make sure proteins get made correctly, get to the right place, and then actually get degraded when they're not in the right place or need to be, are not fully matured correctly. And so we hope that we can do this at a level of molecular detail that will really allow us to rationally control how cells respond to drugs, or potentially in the case of human disease. And so with that, I've sort of hopefully mentioned everyone in the lab as we went through, but I'll just thank the lab. I'm extremely fortunate to have a bunch of amazing PhD students and postdocs from Caltech across all different disciplines, from biochemistry to biology to chemical engineering now. And it's really kind of been a privilege to work with them, and all of this research is not possible with them. We also take a lot of advantage of the really amazing facilities at Caltech and core facilities, particularly the cryo-EM facility. So these are really fantastic and essential resources for our research and many others on campus. And thank you so much, of course, to Chuck and the KNI and you for your attention.
Thank you very much for this talk. I was wondering, for the machines that you're talking about here, are they passive, or do they require any kind of metabolic energy in order to get the proteins in or prevent them from going in?
Yeah, that's a great question. So the ones I've talked about today, we don't think that they actively require energy. So they're not hydrolyzing things like ATP or something like that. The way that they work is they actually you get energy both from interaction with the substrate and because most of the substrates I've talked about today are actually hydrophobic, you get energy from putting them into these hydrophobic bilayers. Because when you have something hydrophobic and aqueous, there's this huge entropic cost to having all of the water molecules organized around it. And so there's a benefit to having it in the bilayer where you don't have that problem. And so you basically balance the energetic cost of moving, or the energetic barrier moving a soluble domain across the membrane, with the energetic desire for these hydrophobic sequences to be in these hydrophobic environments.
So great talk, and I think you've done a great job motivating how these gateways are three-dimensional structures, they're charged structures, and so they require specific orientation in order for things to pass through them. And as I'm reflecting on the early parts of your slides, you show these cartoons of the cell, which I'm familiar with from my youth. But as I grew older, I realized that the cell is really packed with a lot of stuff. It's hard to understand how these proteins can simply diffuse through the interior of the cell, which then leads me to believe that perhaps there's other ways that proteins migrate through and between these organelles, which can be easily as complex as these gateways.
Yes.
Can you comment?
Yeah, so I think the more we learn about the cell, the less I think most people believe that it's just sort of random diffusion happening, because it doesn't quite make sense, because you're right. I mean, the concentration of protein in the cell is about 300 mg per mil, so this is enough to crystallize protein, right? It's crazy. And so I think what we've learned is that the cytosol, so this sort of aqueous space that I draw like this, is not unstructured. So there's localization of some of these things at or near these different compartments where they need to be. And then there's a really stringent pathways by which these are moved around the cell. And our lab thinks a lot about that too, but I didn't talk to you about it. But things that recognize newly made proteins as soon as they are made and then target them to different places, moving them in very directed ways. And I think it's that way that you actually get this movement. But I absolutely agree. I think the sort of random diffusion model it doesn't make any sense given what we know.
So, great talk. Even an applied physics major can understand some of this stuff. But so then we hear a lot outside of biology, we hear a lot about the blood-brain barrier, how hard that is. Could that be also related to charge? I mean, charge, pretty fundamental, electromagnetism. Is it something else, or do we not know?
That's a great question. I'm not as familiar with the molecular details of the blood-brain barrier, but my understanding is it's a little bit more complicated. I think it's probably not just charge, but it's also other molecular properties as well. Things like hydrophobicity and shape and that affect actually how things get through the blood-brain barrier. And that's obviously active area of research on campus as well. There's many people thinking about how to do a better job of getting drugs, for example, into the brain to treat some of these diseases. So.
Hello. Sorry, Jose Gonzalez. I have a question about the charges from the EMC. So you indicated that, it's negative or positive, right? So the other molecules that might be directed to, you mentioned mitochondria or peroxisome. That's one other charge shared between multiple compartments now. Is it possible that these gates or these channels have multiple gates involved in selectivity?
Oh, so I think something like the EMC. I think the gating mechanism is probably roughly what I told you about. So I don't think there's an additional gate. But what is the case is I like to think, I don't know if you remember, the New York Times talked about this over and over again, it's the Swiss cheese model for COVID prevention. And it was this idea that, you had like masking and vaccination and you have ventilation, and each piece is like a piece of Swiss cheese. It all has holes in it, none of it's perfect. But you put them all together and then you get something that's highly protective. And I think that's the same thing that happens in a cell. So there's a selectivity at the membrane, and it's not that great. It's okay, it helps, but there's also selectivity on what gets to the membrane, so transport like we were talking about. And then there's selectivity of what gets inside the ER. And there's quality control, so if it recognizes that there was a mistake that was made, we can pull it out and get rid of it. And so I think you add up multiple selectivity filters together, and you get a highly selective process. There's no one step that is sort of going to be able to be responsible for perfectly selecting out of millions of proteins per minute. It's all of them together.
Hi, thank you. Yeah, I had a question about, you had mentioned that as a future step, the hope is to be able to find some type of molecule like structurally similar to MITCH2 and be able to use that to increase or decrease the activities that we were talking about. I'm curious, for MITCH2, it's specifically on the mitochondria, right? What is the mechanism of like getting it there and having it perform in the way that MITCH2 does specifically?
Yeah, I mean, so just to be clear, what we're really interested in is sort of small molecules that can manipulate its function. So we probably aren't. It would be challenging to identify something that would behave like MITCH2, but I think a small molecule that could bind, in this cavity potentially, that could change its activity, that seems more tractable. But it's a good question. So MITCH2 actually inserts itself. So it relies on itself to get integrated into mitochondria, at least in part. And that's common for many of these molecular gates at different membranes. They kind of get themselves there. And the way that works is that, when a cell divides, there's sort of an old version of the gate that sits at the entrance of the organelle that's still there that can introduce the new sets of particles that are being made. So a common theme amongst these is kind of a weird quirk of like chicken and the egg question.
If you do DNA analysis on some of these cancers that you've identified, do you see differences in the MITCH2 DNA coding or the non-coding regions that affect expression of it?
So the question is, do you see changes in the coding or the non-coding region? Honestly, with the cancers, often it's just upregulation or downregulation. So we don't tend to find, it's very rare to find mutations. Also in sort of other disease models as well, in Alzheimer's disease and obesity and sort of cardiovascular disease, what we don't seem to find are very specific mutations, but actually expression level changes. And they're often from changes in sort of the promoters or the introns that just affect the levels, which is interesting in and of itself, but it's sort of a quirk of MITCH2. It's a little bit unusual that that's the case. I think there's a question in the middle.
I'm sorry. Thank you so much.
Yeah, thanks so much. I wanted to also comment that the cargo itself, or the client, or substrate, whatever you want to consider it, also is subject to post-translational charge modification through phosphorylation or acetylation as examples, that I think is also going to be part of the selection process. It's not just at the gate, but it's what happens before the gate. And what I'm going to propose is that you also want to establish gradients and concentrate your substrates close to the gate. So one way to do that is liquid phase separation, and certain proteins can achieve that. And then the other thing is you've got to unload the cargo once it gets to where it wants to be. And so you can't just have, there has to be a second process that destabilizes the association between the cargo as it's going through and getting to the inside. And that could also be an environmental change in the membrane itself, or through, again, a charge change on the cargo once it's getting to the inside.
Yes, so I think absolutely there's roles for post-translational modifications, and we've been really interested in looking for regulation of particularly these molecular gates, potentially some of the substrates, although for whatever reason, phosphorylation is usually less commonly found in these proteins that have to sit in membranes because phosphorylation is highly hydrophilic, and so it's not going to want to be in a bilayer, a hydrophobic bilayer. But this idea of unloading is really interesting. So one of the things that these gates, and it's consistently found across really evolutionarily distinct sort of membrane protein gating mechanism or molecules, is that they actually narrow the lipid bilayer right at this sort of entrance, right at that filter that I was talking about. And so they make it a little too short to accommodate the client. And so what that means is that you have that driving force for dissociation. So you interact, but it actually won't be favorable for it to sit right here because it would be sticking out into the aqueous environment. And so then that drives it to kind of diffuse away into bulk lipid. And so we think that's part of how you actually get disengagement as opposed to just clogging at all of these sort of complexes. It's something that you see over and over and over again. That's another kind of feature of these molecules.
I'm curious about the single particle cryo technique. What about the role of associated solvent, basically the small molecules around it? You probably need a pretty low pressure when you're taking that scan. And if you lose the smaller stuff around, does that bias the fidelity of the shape that you get?
So most of the things that are surrounding these molecules, so small molecules, but even small proteins, but solvent density and things like this, the sort of one of the pros and major cons of this technique is it's an averaging technique. So you use millions of images and then you average them together. And so if there's small molecules or solvent effects, those are likely to be different in each one of these images and in some sort of random way. And if you average them all together, then essentially that creates your noise. So if you have too much of it, you won't see your particle at all, so that's a problem. But if you have just some, and it's mostly can average away, and the density that you have for your protein, the signal to noise is high enough, then it just becomes part of the noise from your structure. And so in many cases, if we're careful about what the solvent is and the small molecules are there, we can tolerate that kind of noise and it will average out over enough images. But you do have to be very careful that you're not looking at noise and you don't fool yourself. So we think about those kinds of things too.
Give our speaker a round of applause. Thank you so much.